Hello, good evening, everybody. So my name is February. I'm here in Luminocean to record basically the corals here on Banda Islands, and I know a bit about coral identification, and I'm really hoping that you guys can maybe take a few tips or something like that and take it home and then use it for your own coral spawning stuff or for your restoration or whatever have you that you actually need to use it for. So let's. Oh, and this is, by the way, only part one of the presentation. There's about 73 genera that we're going to run through. So, with that being said, let's not waste too much time. So, of course, first of all, we have to describe what even is a species. So we try to identify generally to species level. And sorry, I'm just gonna turn off my video just to make sure that the internet goes well. Do you mind if like everybody else turns off their video? Or anyhow, do let me know if the internet if you can't hear me or the internet breaks or like it starts being slow. Just please do let me know. Or if I'm talking too fast, also please let me know. So generally here we try to describe organisms to species level. So this is what we call the um, how we classify organisms in the first place. So first of all, we start by domain, kingdom, phylum, class, order, family, genus, species. And species is the most specific form of categorization of an organism. So generally, it's a group of organisms that can reproduce with each other and can produce fertile offspring. Of course, lots of caveats with that. Hybridization can occur, especially with corals. So take it what you will, but generally we do try to describe we do try to describe corals to species level. So generally we look at it like this, it becomes a tree of life. So first of all, we start with eukaryotes and then we're animals, and then we have spines and we're also mammals. We are primates, hominids, then here we are Homo sapiens as a species. Then this is for cats. We're quite related, but cats are more related to tigers. Then for corals, there's a completely different offshoot to Cnidaria, and it goes down all the way down to species level. So generally, we try to identify the species level. Ideally, everything should be done via the species level, but it is realistically speaking quite difficult. It is sometimes a bit difficult to guess which kind of characteristics we're looking at. Even like there's several species that really look the same and can only be differentiated by looking at other things like how they sound or what they or little organs that's not really visible to us or even genetics. So generally for field work, it is sufficient to identify the genus. Of course, when we talk about research, the different matter, but generally when we're doing field work, we try to go to at least species level. If possible, genus level, then of course possible, then we do species level. So right now, when we're talking about this, there is currently a shift in what we're actually thinking of species in the first place. So species are always constantly being updated now with molecular information. Um, Everything is getting reclassified, everything is getting reshuffled. There's so many genera, different species, there's families that are being shifted right now because of new research. So right now, the species that and the genera that I'm talking about will be from the World Register of Marine Species, which is called worms. But you can also cross-reference with corals of the world. So first of all, let's talk about what kind of characteristics that we actually look for in for identification. So a coral colony is generally built up of a corallite, which has a skeleton, and it has a live tissue. When it has live tissue, when it has live tissue, then it's a polyp. When we're just looking at the skeleton, it's just the corallite. So the corals, generally they form a bunch of corallites together, and like these generally, we look at corallite and polyp to characterize an coral two species level. So we look at here, these ridges here, and the ridges down here. And then sometimes we look at the tentacles and sometimes we look at the fleshy bits of it. Very rarely do we actually depend on color to identify. So here we have a couple of characteristics that we actually do need to look at. So these are all really zoomed in characteristics. So this is why you do need to like, get your photography skills a bit higher to even like have a look at some of these characteristics. So here, this is like we call it shared walls. That it looks like it's distinctly they're kind of like mushed together. There's no distinct partition between the corallites. The corallites is each single one of this is a corallite. And here, with separate walls, we can see that the corallites are actually quite distinct. They're separate from each other. Like there's a boundary. There's a clear boundary. Then we have no and indistinct walls. Now, these are some of the characteristics that we actually look at. Then here is another key feature to identify some genera. One of them is called paliform lobe. So essentially, like there's a spike of like skeleton in the middle of the corallite. So here, there's a small spike, very small, but it's distinct. Here, there's also a small spike in the middle of it, near the mouth. So this is the mouth. The middle part is the mouth, and there's spikes, skeletal spikes inside. This one is very obvious. And then we can also describe the corallites as having meandering ridges or valleys. Now, this term I took it from the Coral Finder. It's a very helpful term to describe a certain genera as well. So it looks like there's a bunch of mouths in the middle of these ridges, and there's like skeletal valleys that form around the mouths. So when we come to the structure of coral, sometimes we can't see it. There's basically flesh on top of skeleton. It's just like us. So when we have to look at it, sometimes all we see is the flesh. Like for example, like a lot of the bubble corals, like this one that we see here, Fibrophilia ancora, we can't really see the skeletal features. So when we want to see the skeleton, we actually have to walk it out to actually have a look at the skeleton. And these alternate, like if you see this one, the alternating septa actually provide clues as to what the species is. So we walked it with water. We just wave at the water at it gently, so the bubbles won't pop, and you can see the skeleton. So since we're talking about shell spawning at this point, we also want to describe what genera are actually reproducing, and we want to see what exactly that we're looking at is spawning. Because we do want to input this information, we do want to record this information for the purposes of COSMARINDO, and please don't forget to log your observations in the coral log. So for this guide, I've actually put up the known reproductive strategies for the genera that I described. As I said, this is known. It could also be completely different, or some of them just have question marks because no one's actually ever observed it. That coral spawning is something that's like difficult to observe if you don't know when it's going to happen. So most corals are actually broadcast spawners. So they release gametes, just like this Acropora, releasing gametes. So these are egg and sperm bundles because it's hermaphroditic. So each one of those little pink dots actually is composed of both sperm and eggs. Then most corals are broadcast spawners like this. There are other species that are gonochoric, which only either release eggs or release sperm. Each colony has a distinct. Then we have brooding corals that boys release sperm, and the girls kind of catch it, and the girls develop the larvae inside them, and when the larvae is ready, they get basically ejected out. And a lot of corals, some of the corals we actually look at actually do this. So like this is the Isopora. This is the Isopora that we have here. Nabila took this picture, and she observed the sperm release. Now it's very difficult to actually catch brooding coral spawning, especially because the larvae are so small and it looks like that. So what we usually see is actually just the sperm release. So when we're talking about coral identification, it's quite important to look at the growth forms as well. So some of you might be more familiar with this, but we generally describe how the colony looks. So for this guide, I use branching, which means that it's like staghorn type, and crusting and plating, which looks like a sheet on top of the substrate. Then foliose and vase-like, which is basically it forms like little vases, or sometimes they form like sheets off the substrate. Submassive, which is a bunch of columns that are packed together, and it looks like a massive sometimes, but it's actually a bunch of branches together, really thick clumps. Massive colonies form mounds, and they generally are differentiated from encrusting types by the dome that they form on top of the substrate. Though the boundary between encrusting and massive is incredibly thin, because all massive colonies begin as encrusting. Then we have our solitary corals that are usually just a single colony and they are still attached. There are some that remain attached even as adults, and there are most of them actually released from the substrate and then just walk around as themselves. So first of all, we're talking about what exactly is a hard coral, as defined by the tree of life. So hard coral is a hexacoral. It's a coral that actually has 6 tentacles, as compared to corals that have 8 tentacles and are generally considered as soft corals, quote unquote. Now, there are some hydrozoans, which are more closely related to jellyfish, and there are some octocorals, soft corals that actually have skeletons. So both of these have several species that do form calcium skeletons, how we think of hard coral. I did put them in this guide, but you should definitely make a note that these are definitely different than our standard hard corals. So when we're talking about how to take pictures underwater, this is something that is it takes a bit of time to master, and honestly, like not always easy, especially when it's the lighting conditions aren't right, and like sometimes the colony does not want to participate in the process in the first place. So when we look at the pictures that I have here in this guide, so I generally say that the rule is that you should take either two or three pictures. So first of all, you want a wide shot of the colony. You want to see what the colony looks like. So here on this right, the Goniastra that we see here is actually a massive colony because it's forming a dome over it. And then the mid shot, which gives you a relation of how the corals are actually how the coralites are actually packed together. And then the close up macro shot, which actually can see the small features in it. So you can actually see the paliform lobes that I actually mentioned before. It's slight, but very visible. And as you can see, it's quite small, the coralites. So, oh no. Then you know what? Use this guide, use Coral Finder, use Corals of the World, or submit to iNaturalist for any form of help, because this is a learning process for everybody. It took me a while to start learning this kind of stuff. How do I get rid of that? Scratch. So for photography, I corals for identification. So yes, as I said, wide angle colony growth form. And close up, then we want to take pictures of distinct colonies. So sorry, of distinct parts of the colony. So for example, a table, we want to take pictures of the side, the middle, and like the very edges of it. So these are all distinct parts of the colony, and each one can give like very important clues as to what maybe even the species is. Obviously, for me, this is an acropora, but not so obviously what is the species. So this one, if you take good pictures, you can actually keep it as reference. And you can actually show it to somebody who may or may not know what it is. You post it online on iNaturalist, they can have a real actual discussion as to what the species is. So, and it's really important that you take a picture with a scale. I use the ruler. I travel with my little pink ruler. It's been with me in 3 countries. But if you do not have a ruler on hand, use anything that can provide a scale, like carabiner, or maybe even your fingernail or something like that, a full health chart. Use whatever that you can to provide a scale. So when we talk about here, this is how I do it. You need one, a camera, obviously. You need a flash or a torch, and you need a ruler or a scale. So please make sure that you manage your white balancing so you can get the clearest and crispest shots. So when you actually look at this video here, I manage it with a ring light. So this is the ring light that I actually use, and with a TG7. You just screw it on. It's a plug and play kind of situation. It doesn't need to have the strobe lighting or anything. Just keep it on during the dive. And then you can actually get the crispest shots because you illuminate it. You don't need a ring light. You could actually use your inbuilt camera flash, or you could use a strobe if you have that, or whatever you can do. So if you can stabilize your shot, you can focus your shot, you can get characteristics like this. Okay, sorry, Joanna. How do I get rid of that blue line? Quite distracting. So when we want to photograph corals for egg trapping, so this may be not so visible in this photo, but generally, like, you want to take a picture of the the spawn trap with the depth, and then you have a colony wide shot, and then you have your close up shots. Of course, with the scale, rulers are very important to determine different species. So when it's spawning, it's a different situation. You have light here. You have background light. During spawning, you generally don't. So one, either use your torch, or two, either use your flash. Make sure that you keep your white balances properly. When you use read the manual for your camera, it really helps. So you want your depth, you want your time, you want the colony wide shot, and the coral light details. So for iNaturalist, it's a great public platform. I've been talking about it all the time, and like I try to promote it everywhere I can. So you can post any pictures, not just coral. Like if you want to take pictures of fish and then you want to post it, it really helps to post it up, and then you can get other people who are really interested in that fish for some reason to identify that fish, and maybe you can even go to species level with it. So it is actually a really useful way to keep track of the geographical locations of all your observations. And then it becomes like a peer review system, like how you have your journals, but instead it's photographs. So and then after that, you can actually see a map with all your observations, and then you can actually download data using the GBIF system. So and then afterwards, your observation can then be downloaded by other researchers and then be used in the research. So for this guide, bolded words are really important points that actually differentiate this genus from the question with the others. And then it's distinct to this genus. There are always, always, always exceptions to the rule. So always just ask if you don't know. Just to let you know, a lot of this guide is actually what I have come up with. There are some of them that I derived, of course, from Coral Finder or Corals of the World or from other resources, but these are the tips that I have been using, that I have learned to use. I have never been taught properly taxonomy, so this is the way that I have learned. So for Acropora, this is the most common genus for most of you. So first of all, this is your standard Acropora branching stuff. This is some weird digitate stuff. So we always have an axial corallite at the tip, and then we have our radial corallites. So when we say that axial cord is always at the tip and the radial corallites are always in the center, always on the side of it, and they generally always look different. So here we can see here this species has an axial tip. It's very distinct, and the radial corallites that are very different from the axial corallites. So they grow outwards this way. The axial will grow outwards, and the radials will start forming on the side. It's similar to here. So with an axial and a radial. So these are just some more pictures of some weird acroporas as well. So this one formed a table, and this table has strange corallites. There are axial corallites here, and there are radial corallites here. You just need to train yourself to actually say, oh yeah, this is a table. the axial coralites, radial coralites, and you can actually see that the coralites here on the left, which is on the center of the colony, and here on the right, which is the end of the colony, it looks different. So these are actually really important characteristics that you want to see in during when you're trying to photo for ID. So here, axials, the radials look a bit different, and here barely looks like an acropora, but if you squint, there is still an axial coralite, and if you squint, there are still radial coralites. Next is Anacropora, which is still in the family Acroporidae. It is related, and it kind of does have a distinct it does have an axial coralite, and it does have a radial coralite. It sort of looks like it, but if you see, like, it doesn't project. It doesn't look full. It actually looks really smooth in comparison to Acroporas. So they generally come in more bushy forms, and then you're gonna have to actually look for it. Just get used to Anacroporas. Next is Ceratopora to distinguish between Ceratopora and Anacropora, because both of them actually look kind of similar. They're very sharp and pointy. But here, if you see, it looks really Anacroporas look rather smooth in comparison of Ceratophoras that actually look kind of fuzzy, because there's a lot of coralites here in the middle of it. And these coralites tend to kind of be flush, and they also form distinct lines. So they actually line. The coralites form in a linear fashion. So that's a distinguishing feature of Ceratopora, is that the coralite always form in a line. Okay, all right. So okay, for Styloporas, they kind of look like a Stereatopora. Some of them really do look like Stereatopora, not here though. So they form these kind of clubby stuff, and they form very different morphologies depending on the depth. So here on the left here is a deep water specimen. The middle one is a mid-water specimen, and on the right is a shallow water specimen. They may be the same species, they may not be. We don't know. I don't know, at least. Maybe there may be genetic studies that actually do require this. So Stylophora can be distinguished by this spike on top of each coralite. So like these spikes are very much a giveaway of what a Stylophora is. So they form clubs and they form spikes. So for Isopora, um, on the right, we see Isopora pallifera, which is very, very distinct. It has gigantic clubs, and these clubs have coralites that kind of look like bitten donuts. And this is very common here, Isopora pallifera. Now, there's another species that we actually need to look out for in Isopora as well, and it's Isopora brueggemani. Brueggemani looks like Acropora at first glance. At most glances, it still looks like Acropora because it looks like it has an axial coralite. It also looks like it has a radial coralite. But if you look at the side of the branches itself, you can actually see that there's a few axial looking coralites as well. And this is very distinct. Isopora brueggemani is very distinct once you actually have seen it and you understand it as Isopora brueggemani. You will see it everywhere. You will not mistake it as Acropora once you actually know. Varieties is, I would say, quite widespread, but also usually easy to distinguish, but sometimes not so easy. So we have our varieties, massive varieties here on the left side. This is our massive varieties, your typical ones. They have very, very tiny coralites, about like 1.5 to 2 millimeters in diameter. So colony shot, and then small individual coralite shot. Then we have our branching types, which this one is a variety cylindrica. So we call them also finger corals. So sometimes the finger corals also have very tiny coralites, but also some species also form like deep corallites, or sometimes they have like meandering wall type situations. And that's why it's really important to take a picture of the branches, or like individual corallites with a scale, because otherwise you won't be able to measure the size of the corallite and the size of the crevices. So here we have Varieties roost, which is a daytime spawner here. So this is, well, it kind of looks like either Porides or Montipora, but after you kind of get used to what a Porides actually looks like, it kind of gets easier. It kind of looks like drip candle wax from outside. Then there's a lot of different species that actually start looking like Montipora, which kind of gets a bit confusing, which is why next I will talk about Montipora. So first glance, if you don't really know what this is, you also guess probably Porides, but it is definitely Montipora because it has like these random looking bumps everywhere. So these random looking bumps are quite distinct to Montipora usually, and they look really random. For Porides that have bumps, it doesn't look as random. So it's a very thin line, and there's even a species called Montipora porides because it looks like porides, but it's actually Montipora. So this is another branching kind of Montipora. It has these really like these random stinules on it, and this is what it looks like from outside, from a wide angle. And here is another example of Montipora. Underneath all these tentacles, you would actually see that it has like these random bumps. So it has a really complex skeletal structure, and generally incredibly varied. So it's always better to take a picture of it and ask who about it until you actually can kind of tell. And even now, I still mix my varieties and Montipora. So Goniopora is this one, I would say, is relatively easy to identify, and it's generally also everywhere. So most people would at first glance think that it is a soft coral, and it is not a soft coral. It is definitely a hard coral. If you waft it, the tentacles retract into the skeletal body. And then here is what it looks like retracted. This is of the same colony. Now, if you see, sometimes there will be a really surprisingly very fluffy looking varieties. It looks like a varieties, but it looks really fluffy. Sometimes it is Goniopora because it is in the same family, Poritidae. But generally, you can distinguish between varieties and retracted Goniopora by having these little flower patterns around the coral colonies. Like Goniopora generally have these. And then like this will come in handy later. So this is the skeletal structure of Goniopora. So for Alveopora, it looks quite similar to Goniopora, but the distinguishing feature is that it has 12 tentacles in comparison to Goniopora's 24 tentacles. So a 12 tentacle Gonio Alveopora sometimes is very obvious like this. Other times, not so obvious like this. It just looks really fuzzy. But if you actually like put your ruler to it, it is solid. It is really solid. You waft it and it looks like this. Now, you can actually see that it kind of looks really different from the goniopora. It looks like the holes look actually generally larger and it doesn't form that flower pattern that I mentioned before. And it's because here the skeletal structure is actually a lot more porous when it comes to alveopora compared to goniopora. Here's another picture. It generally looks more compact because the tentacles are closer together. Okay, we are now at medium level. Ooh. If you guys have any questions, just drop a chat, drop a message, and then I'll have a look at it later. So for Hypnophora, I call this the lemon squeezer coral, because they tend to have really sharp ridges. So these ridges are quite distinct to Hypnophora. This is Hypnophora excessa that tends to have ridges that look a bit oblong. Some of them are cylindrical, some of them are long. And then this is Hypnophora microconos, which form generally circular lemon squeezings. Then this is Hypnophora rigida. Now, Hypnophora rigida is the only branching type and is very distinct once you actually look at the surface ridges. The surface ridges are incredibly distinct to Hypnophora. Then, oh, and don't forget, like there are tentacles generally in between ridges, but do not mistake this for Galaxia. Galaxia tends to have a callus that actually raises out above the encrusting base. So this callus is raised, and then there's a mouth inside, and then there's a ring of tentacles, sometimes there's not, depends on the coral's mood. You waft it, the tentacles sometimes retract, sometimes they don't. And then this is the branching form of a galaxia. It looks spiky, it's quite sharp as well, but very brittle. You tend to find this in the same kind of environment as you see Anacropora. And you can actually still see that the calluses are actually still raised, and they actually form like still this sharp spikes. Sometimes they have tentacles, especially at night, but during the day they don't usually. Now, lobophilia is commonly known as grain coral. It's quite distinct. It has a very meaty look to it, and they sometimes form like these these meandering ridges, and it looks really generally like these meandering ridges tend to be very thick, and the valleys also tend to be quite deep. It looks like a bunch of sausages, or like I guess people call it brain coral for a reason. So they come in both massive and submassive, and when they're babies, they can form in encrusting forms. Oops. So generally for this species, for this genus, like the thickness of the walls and the ridges and the texture of it, and like sometimes you can even see the skeleton, or like even the mouth placement. They can help to identify it to species level. I can't, but I'm sure somebody can. So for Pectinia, the most common form of Pectinia that we see here is Pectinia lactuca here in Banda. So they form incredibly deep, deep ridges. So the walls tend to be quite thin, very thin walls, and very deep ridges. So this actually forms the mouths are like quite deep inside. This one as well. Sometimes the ridges aren't extremely orderly, and they can turn out quite random, like this specimen. Or like this specimen as well, it also looks quite random. So sometimes some species they form kind of well Nick, my previous dive instructor calls it French fry shapes, because I guess he was hungry. But generally, like you can see that these actually have a thin layer of flesh and are quite fragile as well. So if you start to see these really deep valleys, then you would actually consider this as pedia. Next is Samopora. It is deceptively smooth, I would say. It tends to look really smooth, especially when you see here. But if you zoom in further, you zoom all the way in, you can actually start to see that there's a pattern, a very, very, very tiny pattern. So I call it rice coral because it forms patterns of like the grain of rice and around the mouth. So here you can actually see a single mouth here, and these little, little, little rice patterns actually distinguish it as Samocora. So it looks deceptively smooth, but when you zoom in, as you can see, it's really tiny by the scale. It's very patterned. Now this is a bit controversial right now. It's a Cosinaria. Cosinaria, well, it used to be a different genus. It may be the same, maybe Samocora now. I'm not quite sure what is the rule at this, but you can actually start seeing the little rice patterns as well in the valleys of the walls. So if you get that, like ridges with little rice patterns, then you can call it either Cosinaria or Samocora. Somebody's gonna pick a fight with you, that's fine. So Steloceras in first glance also looks like every other um, massive colony, but if you actually have a look-see really close with a ruler, there you go. So generally they form corallites that are about like 4 to 5 millimeters in width, and then you can actually start to see the little blades go in, the septa go in, and they have shared walls. Looks like it at least. And then they have little dots in between these little blades that lead into the mouth. So these little dots are actually tentacles. And during the day, very, very distinct. If you start seeing these little tentacles and little dots in between the blades, then that is Steloceras. And there's only one species of it, and that is Steloceras myarius. So here we have the massive and encrusting growth form of Osteopora. Osteopora is surprisingly related to Acropora somehow. I am not here to judge, but it can get really big, and my friend calls it googly-eyed coral because you can actually see the little mouths actually start end up pointing everywhere, and like if you were to shake googly eyes and then just like lay down, these little coralites really spread out and they go everywhere. And these coralites form like little ridges that go down. So if you can see these ridges, they actually go down in a kind of like straight pattern down. So it goes from the mouth and goes straight down in a little ridge. Then we have our strange encrusting forms here. They do get like this sometimes. I thought this was a Montipora first until I actually realized that it's actually Osteopora. And I forgot to put the very zoomed in picture of it, but you can check it later on iNaturalist. Then Diploastria heliopora. This is probably the oldest lineage of corals. This is like the coelacanth of corals. It's extremely ancient. I think it ages to like 35 million years ago or something like that. And it's very, very, very distinct. So normally they come in this kind of brown, creamish brown color, and they form little low mounts that are like volcanoes. And they have a kind of beaded surface like the Osteopora, but this is like lower, and you can actually see the inside of the mouth usually. And they form these little lines all the way down to the base of it. And then in between, there will be other little baby coralites as well being formed, and they can get very, 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 very large. Oops. So for Cyphastria, also, once you get the hang of it, it's very, very easily identifiable. Cyphastras generally have like these ridges that go out, like these septal blades that go out, and then it goes into this kind of like rough area in between the coralites. Like this area of the coralites, tends to be rough. It looks rough. It looks sandpapery. There's little spicules in between the coralites. You can see it here as well. So it looks like any other massive colony yet again, but then when you actually have a look at it closely, it has this little like star-shaped thing, and in between the stars, there's um there's a rough area. So there's actually three here. Like you can actually see three shots of the same colony. It looks different, but also the same because you actually see the little stars, and then you actually also see the little areas in between that have a little dots. So the only exception to this is the Cyphastria decadia. Cyphastria decadia is the branching form of Cyphastria, and I've not seen it here in Banda, but I have seen it quite frequently in Lombok. It looks Cyphastria like a regular coralite of Cyphastria at first glance, and then you get stare at it, and then there's no sandpapery texture at in the side of it. Generally there is, but this one may be a bit too vague or covered by tissue to be able to see it. But you can actually see the similarity of the shape of the coralite. All right. Then we have Pachyceras. Once you have an eye for this, it becomes quite easy. It's also known as the elephant coral because they form, I think it's because they look like elephant skin or something, or elephant ears. So the Pachyceras, the folios Pachyceras form concentric rings from a center. So they get formed, but and these rings don't usually break. And then the massive forms form columns just made out of concentric rings as well. Uh, so this is what it looks like zoomed in. So it looks like it doesn't look like it has any mouths or anything like that, but but there's like lines that radiate out with the concentric rings radiating out from the center. So we're starting to get a bit more challenging here. So this is Gardneroceras. So there's only Gardneroceras planulata in this species, so in this genus, so it does make it a bit easier. It starts to get a bit difficult here. This is Gardneroceras planulata, and it looks honeycomb coral generally. It looks really deeply excavated. It's like looks like it's been scooped out, and then it has a little star here in the middle of it. And then this little star, and in between the corallites, there's generally a very pale yellow or whitish border, and generally like these little septal blades, they don't cross. So this border is quite distinct, and the septal corallites, the septa of the corallites don't tend to run. So Pavona is where we start to get a bit more difficult. So Pavona is there's no quick or hard rule to it, unfortunately, and generally we only start to look at the septacaustic. So they tend to run. Now, if you see this and you compare it to the last one, it tends to look quite similar. Like compare this with Gardneroceras, it is kind of difficult. Gardneroceras tends to be a bit more scooped. This is less scooped. That is very relative. I know the description. But if you see here, the septal blades actually run to the other side. And in the center is the Pavona. This is Pavona look. Now, if this is the and massive, this is a massive form. And if you see here in the foliose form, it also forms this flower pattern. This is the Pavona look, this flower pattern. So Namila calls that mate. So you can also see here this radiating septa from a tiny mouth, and it runs to the other side of the other corallites as well. So, okay, hang on. Let me have a look at where I'm supposed to go. All right. We can stop soon and then have the next part later on. So for this Leptoceras is you also need to actually kind of recognize the look. And it tends to sometimes it tricks you and it may or may not look like Pavona. But it also has the septa that run from one corallite to the other. So it runs and it runs and it runs. But Leptoceras has something that Pavona doesn't, and it's this squiggly line. So they run, but then they ran into random squiggly lines all of a sudden, like this one here. This squiggly line is very distinct to Leptoceras usually. And here you can see it, like these two are both Leptoceras. This is also Leptoceras, and it also looks like a Pavona. But if you see, compared to like this and other Pavona specimens, you can see that there are random squiggly lines that come out of nowhere. Like this one, squiggly line, here, squiggly line, random. So Leptoceras is, for me, is squiggly line coral. And then you get this, which is Leptoceras fabricea, which is incredibly fragile and usually incredibly small. And they kind of look like rubble. So this is generally why I tell people not to put their fins on the reef because it can look like coral because it usually sometimes is. And Leptoceras fabricea, they tend to like generally deeper environments or at least more turbid environments. And they have these kind of radiating things could come from the center and very few squiggly lines. But this is very distinct that this is Leptoceras fabricea. Okay, starting to get a bit rough here. This is mycelium. Compare this with other folio stuff, it tends to get a bit difficult. So here we actually start to look at, well, they look like little noses radiating out from the center. So each one is a nose, and in between, and like stuffed up to every nose is like a hairy nostril. The corallites tend to actually have the tentacles like all just out like that, puffy like that. So this is Mycelium robococci that tends to look quite smooth. In comparison here, we have Mycelium elephantotus, Now this looks more rough in comparison, I would say, but they still have this distinct um, nosy look to them that radiates from the center to the outside. And then this mess is Mycedium mancaoi. We have a lot of it here in Banda. They also have this incredibly nosy look to them. Sometimes juvenile specimens look incredibly different, and some specimens just don't particularly want to obey rules. There's only a few things that actually give it away, and this is why it might be difficult between this and another two that I'll talk about later. So here for turbinaria, well, maybe I should put here first and leave the rest for the second part. Yeah.